Pests destroy up to 40% of global crop production every year. We typically fight them with synthetic pesticides, but insects and fungi are rapidly evolving resistance, rendering our current chemical arsenal increasingly ineffective. A highly effective alternative already exists in nature. The fungal order Hypocreales naturally colonizes agricultural plants and secretes specialized chemical compounds that kill, deter, and inhibit invading pests. The instructions for building these chemical weapons are hard-coded into the fungal genome. They exist as biosynthetic gene clusters, or BGCs, contiguous blocks of genes that work together as a single operational unit to assemble complex molecules. Sequencing the DNA of these fungi is no longer the difficult part. The bottleneck is translation, taking a newly sequenced genomic blueprint and identifying the exact physical molecule it produces in the real world. Bridging this gap between genetic potential and chemical reality is a necessary step for engineering sustainable, biologically based crop protection. Researchers mapped the genomes of 82 Hypocreale strains, identifying over 5,000 distinct biosynthetic gene clusters. On the right, red markers represent predicted products the fungi can make. The sparse blue markers represent products actually detected. The math is staggering. Approximately 80. It combines genomic mapping, chemical analysis, and synthetic biology to force these silent genes to reveal their outputs. We do not need to find new genetic clusters. We need to figure out how to make the cryptic ones we already have start speaking. The first step is to attempt to shock the fungi into action. Researchers use the OSMAC approach, one strain, many compounds. By cultivating the fungi across drastically different nutrient conditions, they attempt to mimic ecological stress and trigger those silent genes. This creates an immediate, massive analytical bottleneck. To see what the fungi produced, researchers must process over 450 distinct extracts through untargeted liquid chromatography tandem mass spectrometry. When the data comes back, many detected compounds do not match any known reference spectra in existing chemical databases. They are completely unidentified. Scientists translate the mass spectrometry data into molecular networks, grouping unique fragmentation patterns to organize unknown ions into related structural families. These chemical networks are then cross-referenced against genomic data, mapping the unknown compounds directly back to enzymatic functions encoded by unverified gene clusters. Molecular networking creates a predictive map. It traces an unexplained physical molecule back to a highly probable genetic origin. Let's test this map on a specific molecule, dithioceicoemestrin. It is a trioxopiperazine with known antibacterial properties, and researchers detected it uniquely in the extracts of the fungus Acanthomyces muscarius. The molecule's biosynthesis pathway was completely unrecorded.
But based strictly on its structural features, bioinformaticians proposed a 20-gene candidate cluster, dubbed the DSE cluster, found within that specific fungus's genome. The molecule contains an epoxidized ring and sulfurized skeleton, accounted for by cytochrome P450 enzymes DSEH and DSEJ in the cluster. More critically, the pathway requires a bicycle phenylalanine intermediate. The cluster contains gene DSEA, a synthesis built to physically lock onto a phenylalanine molecule. The bioinformatic model fits perfectly, but a theoretical fit is not physical proof. To confirm the DSE cluster actually builds dithioceicoemestrin, researchers had to force the fungus to assemble the molecule using traceable parts. They achieved this using stable isotope feeding. The scientists prepared a batch of the amino acid phenylalanine, but they replaced its standard carbon atoms with carbon-13, a heavier stable isotope. They fed this uniformly labeled phenylalanine directly to Acanthomyces muscarius during cultivation. The carbon-13 acts as an atomic-level tracking device, allowing researchers to follow the amino acid as the fungus metabolizes it. The mass spectrometry chart shows the unlabeled control produced a standard peak at 550. Extracts from the carbon-13-fed fungi shifted precisely to 559 and 568. This 18 Dalton shift proves two labeled phenylalanine units were incorporated directly into the dithioceicoemestrin molecule's core. By tracing atomic weight from the raw ingredient to the final product, the researchers permanently linked an orphan molecule to its previously uncharacterized gene cluster. Isotope feeding requires one crucial condition. The native fungus must actively express the gene cluster in a laboratory environment. For the vast majority of silent BGCs, environmental elicitation completely fails. When a fungus refuses to activate a gene cluster, scientists bypass the organism's native epigenetic regulation entirely, using heterologous expression. Researchers physically extract the cryptic gene block from the fungal DNA. They codon-optimize the sequence and insert it into a high-copy yeast plasmid, specifically Saccharomyces cerevisiae. To ensure the foreign fungal enzymes function correctly, they engineer the yeast host with a specific modification, a promiscuous phosphopantotheniol transferase. This accessory enzyme acts as a universal starter motor for the inserted gene cluster. The engineered yeast acts as a blank, highly controlled biological factory, completely stripped of the complex evolutionary silencers that keep fungal dark matter hidden. To test this factory, researchers isolated gene G9547, an uncharacterized cluster from the fungus Simplicimula, and expressed it in the yeast along with 2 accessory genes. The wild-type yeast control shows a flat line, but the engineered strain produced a distinct new peak at exactly 350.2484. Mass spectral fragmentation identified a novel meroterpenoid, a quinoline alkaloid with a farnesyl moiety. The yeast successfully manufactured a new molecule on command. We now possess a systematic method to force silent genes to yield their products. Science now has a viable path to harvest the 80% of fungal metabolites previously hidden from discovery. By integrating phylogenomics, mass spectral networking, and synthetic biology, we can translate raw genetic potential into physical chemical tools, establishing the foundation for a new generation of sustainable biopesticides.